So, good evening, all. We are back to our fifth session, um, which is about preclinical research. So, basically, this workshop was designed in such a way that we are going to talk about everything from basic research till clinical. And in addition to that, soft skills was one another session which we plan in between. So today's session is about preclinical research, and we have got today with us founder of Cyro Research Private Limited, which is a contract research organization which involves toxicology, pharmacology, microbiology, and biotechnology work. Uh, Mr. Anand Rahigude, sir, who is the founder of this Cyro Research Lab, he is actually looking after all the um, as a founder looking after all these activities which comes under preclinical. He is the responsible his responsibilities of technical director, and he is a chief executive officer of Cyro Research Private Limited. If we look at his educational background, he completed his pharmacy degree from Rahigude Institute of Pharmacy, Nare, in 2010, and then he done his masters in pharmacology from Rahigude College of Pharmacy, Vadgaon. So just one second, I will. Yes, sir is there. And after completing his masters, he has also contributed in different research papers, more than 8 research papers with very good impact factors. And he has associated with many preclinical research and contract research organization for more than 13 years. During this time, he has worked with regulatory toxicology and pathology department as a study director, and was involved in designing, planning, conducting, and reporting of the toxicity profile of the drugs, medical devices, biologicals, vaccines, agrochemicals, and chemical. So during his time, he was also associated with preclinical pharmacology and drug discovery. So Mr. Anand is actually a well experienced person in the preclinical. He has gone several trainings in GLP, ISO 17025, and ISO 9001 during his regulatory research work. So after having contributing for more than 8 years, he decided that he will establish his own preclinical research lab, and he started Cyro Research Private Limited in 2020. In addition to doing, founders work at Cyro Research Lab, he is also associated with different reputed biotech research organization as a research consultant, same like what he is doing with Swalai Biotech. Simultaneously, I've seen his contribution to his previous colleges. He is associated with Seagar College of Pharmacy, taking workshops, then delivering sessions, and so he's contributing as an alumni to his college as well. So with this brief background, um, I like to welcome sir, and discuss about preclinical, discuss about what things Cyro Research Lab is doing. So over to you, sir. Mike, I think your mic is unmute, sir. Yeah.
Thank you, sir, for such good description of for myself. And it's very happy to conduct this session for students or anyone who is listening to this presentation. So rather than wasting a time, let's start with uh, whatever the major topic for this description, these sessions. So basically, my motto behind conducting preclinical research is the uh, I really want to get involved in novel drug discovery, and that was the first thing that hit me to get in the area of preclinical research. So let's start. What is exactly preclinical research? What is its foundational things, and how it goes for uh, for different products and different uh, areas of drug discovery. So, every pharmacy student may know that there is a drug discovery pipeline which goes as identifying the target, uh, either receptors or enzymes that may exert the pharmacological actions, finding the lead compounds that act on those targets, and then going through high throughput screening and finding the exactly efficacious molecules that involve for efficacy of that compound, and then preclinical efficacy testing, which involve the confirmation of pharmacological action of those compound, then evaluating toxicity of those compounds uh, to get those product in the clinical trials. So basic rodent data and non-rodent data is required for the clinical trials in phase 1, phase 2, and phase 3. So once this toxicity and efficacy data is completed, then and only then we can get into clinical trials for novel newly uh, synthesized molecules. And after that, post marketing surveillance that everybody know about pharmacovigilance is ongoing process during the life cycle of that compound. So, what is exactly preclinical research and what is its importance? So, preclinical research is a part where, as we spoke on last slide, that from high throughput screening to the preclinical toxicity testing, this is the part where exactly preclinical research work is conducted. And its major importance is to get understand about the pharmacological properties of that molecule along with its mechanism of actions, how it gets absorbed in the body, what is its pharmacokinetic properties, what is its toxicokinetic parameters, how is there any kind of side effects occur due to those molecule. So to evaluate each and everything related to the that molecule, the we can say a basic uh, basically basic part of everything of that molecule, that is which receptor it activates or inhibits, and what is the exact pathway it follows for its pharmacological action. So all this kind of research is a basic research, and which is involved during the preclinical research activity. So majorly we consider that preclinical research part is a testing in animals, but it's not just testing in animals. There are much more methods involved in preclinical testing, where different kinds of in vitro assays in different kind of standardization parameters are involved to get uh, everything uh, reliable and good data for those compounds or molecules. So, animal models in preclinical research is a major part of a novel drug discovery pipeline, and these preclinical methods are required to be studied during the process of novel drug discovery, and which helps for student to get uh, a regularized part of drug discovery industry. So, what is the significance of preclinical research? So, basically, it is a crucial step in drug development where safety and efficacy of those uh, novel molecules get evaluated, and basically, uh, every data uh, generated from these studies is helpful for evaluation of safety of those molecules while going in clinical trial, phase 1 clinical trial or phase 2 clinical trial, where safety doses can be identified depending upon the rodent based study data, which will help to extrapolate the doses that can be utilized in humans. And in a phase 1 clinical trial where healthy volunteers are involved, we can evaluate those doses for the safety. It basically involves uh, efficacy evaluation and safety evaluation, and giving us insight about identifying risk and benefit ratio. So, if drug is a very novel for a very novel disease, or we can say cannot say diseases are novel, but rare diseases, and there is no uh, marketed drug is available for that molecule currently. So we can identify, and but the developed drug is having a toxic effect. So at the current trait, the drug is beneficial to treat the disease, but having some side effects or toxic effect which can be prevented or which can be controlled by using another medication. So it is a beneficial ratio or beneficial part of that molecule. But there are several molecules in the market, and we have developed a new molecule which is having a best activity apart from those molecules that present in the market, and having a greater side effect than the molecules that present in the market. So in this case, we have high risk ratio, and though these molecules cannot be entered in a clinical trials due to its higher toxic effect compared to the uh, previous molecules present in the market. So that is the basic significance of preclinical research. Uh, so basically, conducting preclinical studies have three stages. First is in vitro stage test where high throughput screening and everything goes on. Nextly, animal models, that is in vivo tests, and evaluating other drugs activity like pharmacokinetic, pharmacodynamics, toxicokinetic, toxicity testing, and overall biological activity of that drug. So metabolism and other parts are also involved in evaluating drug activity during the preclinical research. So, at the first stage, what is in vitro cell line study? We will discuss about in vitro part first. So, what is in vitro cell line study? That this is the latestly involved in vitro method where either primary cell lines or cancerous cell line, we call it as a immortal cell line, or fibroblast type of cell lines are utilized for the evaluation of uh, pharmacodynamic effect of molecules, cytotoxic effect of that molecules, and other related actions. So, in case of drug discovery, in vitro cell line studies are vital in the drug discovery process, where it allows to evaluate safety and efficacy of the product along with the mechanism of action of that product. So depending upon the designed in vitro test method, we can evaluate how those molecules exactly act uh, after administration, and what is the exact mechanism of action. Secondly, high throughput screening is a major part where multiple drug candidates are synthesized at a time, and we cannot utilize them directly into animal models because they might have toxic effects, do not have any efficacious action, and maybe they are uh, just kind of molecules that they are produced by medicinal chemistry but do not having any kind of action. So to eliminate those kind of molecules and to minimize the number of animals in the next study, high throughput screening is required. And in these cases, cell line study is more important because it can uh, screen more than 1,000 molecules at a time, and we can prioritize the lead molecule from those 1,000 molecules which can which can go further in preclinical testing, where they will be evaluated robustly for their efficacy and toxicity. So basically, these are the mechanistic studies where it provides essential preliminary data that is majorly required for animal studies as well as for clinical trial. We majorly say that is this is cost effective and methods, but considering the uh, evaluating more number of molecules like 20, 50, 100. So if that in that cases the methods are very budget friendly and it will help to control the total cost of the whole experiment of drug discovery. So ultimately it will help to reject the molecules that are not active or maybe toxic during the testing, and it is very beneficial to get only efficacious and safe molecules to the next stages. So there are different cell line tests that are performed for evaluation of safety and efficacy of molecules. These are some tests that we conduct at our Scio Research Private Limited, and basically these are cytotoxicity assays where every medicinal substances, medical devices, or any substances that came in contact with humans need to be tested by cytotoxicity assay, which we majorly performed on L929 fibroblast cell line as per ISO guideline. Secondly, proliferation assay. So uh, some compounds have uh, effects that they proliferate the cell, that means they increase the cell number, and which is required in some conditions like anti-aging activity or neuro regeneration action. In that cases proliferation is required, where increase in cell number is a good activity. So these kind of proliferation assays we can perform. Apoptosis assays are required to be conducted for cancer uh, anti-cancer compound evaluation, which will ultimately help us to evaluate apoptotic pathways that involve for the cell killing. Apart from that, anti-aging assays that we can age the cell using some oxidative stress making compounds and evaluating the decrease in oxidative stress marker in those cell by in presence of our compound. Anti-adipogenesis assay using 3T3L1 cell line. uh, Glucose uptake assay in case of diabetes. We can conduct this in vitro cell line based assay where glucose uptake by cell will be measured, and if it is getting uptaken by muscle cell or liver cell, that indicates that those compounds are good in terms of insulin mimicking effect. Osteogenic assay, which in which case bone growth will be detected. drug absorption models are there. So basically, if you are utilizing some eye drop for retinopathy or related to conjunctiva. So in that cases, we cannot evaluate the pharmacokinetic effect because the absorption concentration will be a very smaller one. So in that cases, we can we can evaluate the absorption of those compounds or molecule by cell-based drug absorption method, uh, where uh, we can evaluate the absorption of drug by passing it through the 2D layer of cell surfaces and evaluating from another way. Similarly, cell-based colon absorption models are there, which ultimately helps to evaluate the colonal absorption of some drugs. So, uh, if there are some formulation which can be only released in colon depending upon the pH and stability of those molecules in stomach and other part of intestine. So depending upon that, if those drugs get absorbed from colon, then only it is efficacious and exert their activity. So these kind of colon based absorption models are also there. Cell adhesion assay for probiotics. So majorly probiotics get adhered to the colon and exert their effect like production of SCFA, maintaining gut liver axis. So those probiotics are getting adhered to the colon or not is a major part where some probiotics uh, are included in probiotic category because those are lactobacillus. But every lactobacillus cannot be a uh, good probiotic if it is not getting adhered to the colonal cell. So those kind of evaluations can be conducted at Scio Research Private Limited. Secondly, scratch assay in case of uh, wound healing evaluation of by in vitro method. There is a scratch assay that can be performed where we can evaluate the closure of wound and rate of closure of the wound. Apart from these cell line based assays, there are different assays so which can be conducted by in vitro method. So some enzymatic inhibition assay that we can perform, like amylase inhibition, glucosidase inhibition is required to be conducted in anti-diabetic activity. Tyrosinase inhibition to evaluate the uh, melanin pigmentation inhibition, uh, or increase in secretion of melanin. So those kind of assay can be conducted depending upon the using tyrosinase enzyme. Lipase inhibition is required for anti-adipogenic activity. Membrane stabilization majorly conducted for evaluation of anti-inflammatory activity of molecules. Some plant molecules have antioxidant activity, so evaluation of in antioxidant active potential of those molecules it can be conducted by different in vitro antioxidant parameters. uh, Platelet aggregation, so thrombus formation is the uh, major majorly involved during heart attacks and other complications. So, evaluating drug molecules for platelet aggregation or thrombolysis can be conducted uh, to get efficacy of those compound against these kind of disorder. And his cell glucose uptake is a very basic uh, method to evaluate anti-diabetic effect, like we have spoken previously, insulinomimetic activity of compound. So these are kind of in vitro tests that we can perform or you can perform at your end, where uh, basic screening can be conducted for those molecules. And currently, animal studies required these kind of in vitro testing previously conducted, so that IIC committee or CCSA will allow to conduct preclinical trials for those compounds that are already tested in vitro method, unless and until there is no specific method mentioned for any specific disease that can be evaluated by in vitro assays. So after in vitro testing, molecule gets into next pipeline where it is going through in vivo drug testing procedures. So first, rodent based in vivo drug testing can be conducted, required to be conducted for evaluation of drug efficacy. So basic pharmacodynamic action of that compound need to be proved. Secondly, dosage determination. So we can conduct a dose dependent study where we can evaluate the efficacy of that molecule is getting sealed at any point. So drug dose response curve can be established by using in vivo based study, and depending upon that dosage we can extrapolate the dose for humans and which can be tested in clinical trials. Thirdly, uh, side effect monitoring majorly involved where different kind of toxicity studies can be conducted, and which organ can get affected due to those molecules can be evaluated at the primarily stage. So depending upon that we can extrapolate the side effects as well as how to control those side effects or toxicity of those molecule can be uh, evaluated from the basic of this kind of testing. And majorly animals, majorly rodents are utilized for the evaluation of this kind of activity is due to the rodents have a very close similarity, genetic similarity with humans, and having a close anatomy structures of every organ that with human, and therefore these animals are identical. Preliminary model that is identical with humans and can be utilized for uh, extrapolation of different things from animal study to the human study. So, why animal models should be used in research and how it can be used? So, basic is study disease mechanism. So, first part where we need to evaluate how disease progresses and how we can control using our treatment. So, every disease is having a very complex action, and depending upon the its different pathway of activating diseases, we can choose the substance that can induce the disease. So depending upon that we can design the experiment where we can uh, choose inducing agent depending upon the mechanism of that disease, and which help to evaluate the exact mechanism of action that uh, we have uh, what we can say that we have evaluated during in vitro study and confirming it by this method. Secondly, testing of new treatment is required and pharmacodynamic activity of those molecules can be evaluated by the animal model. It also helps to evaluate drug behavior inside the body. So pharmacokinetic evaluation is major requirement factor for animal model. Majorly used animals are rat, mice, rabbits, guinea pigs, and hamster. Along with that, large animals are utilized for evaluations like primates, and in some cases sheep and goats also used. Second major utilization of animal is required in case of toxicology, where we need to assess the safety of those molecules or compounds that are having a good efficacious property in case of drugs. So if those at first stage when we are conducting efficacy or pharmacodynamic study, the potential candidates who are having a good activity compared to the standard or marketed formulations, those compounds need to assess uh, their toxic effect if there are any, and along with that it helps to evaluate the mechanism of toxicity of those molecules. So are those molecules are affecting our liver or kidney or heart or any other specific organ, or just having a nausea or vomiting like effect due to extreme acid secretion, or those drugs may do not have any colon absorption after oral administration, it is getting eliminated as directly from the body. So those kind of evaluations can be done during toxicology and toxicokinetic parameters of evaluation in animal study. So toxicology majorly have regulatory guidelines that need to be followed during the evaluation, because those are worldwide approved guidelines for evaluation of toxicity, and just not for drugs or novel drugs. It is required to it is required to conduct toxicity evaluation of any molecule that will be in human contact after getting into the market. And major ethical consideration is utilization of number of animals for conducting safety or toxicology testing. So, different regulatory guidelines that are worldwide used for evaluation of toxicity of molecules, drug molecules, or other chemicals is OECD guidelines, ISO guidelines for medical devices, ASTM and WHO guidelines for uh, vaccines and other biological materials. Indian pharmacopoeia, US pharmacopoeia, and Indian standards are indicative for specific drugs. It's like in case of Indian pharmacopoeia, biological vaccines and sera need to be tested by using abnormal toxicity test or undue toxicity test. United State Pharmacopoeia allows to evaluate the packaging materials that utilized in pharma companies by using biological reactivity tests, and Indian standards allows to test the uh, soap and related materials uh, for their local irritation or skin related toxicity test evaluation. There are other agency like EPA. EPA involves or make guidelines for evaluation of toxicity of agrochemicals or fertilizers and biofertilizers. So they have a different guidelines for evaluation of those material. FDA, MHRA, and EMA make guidelines for evaluation of toxicity of medical substances, medicines, and uh, specific chemicals that can be utilized for uh, prevention of diseases. And there are other guidelines like ICH, DCGI, FSSAI, and Ayush, along with their CCRAS guidelines, which uh, defines how to conduct a preclinical testing for evaluation of safety and efficacy of the molecules. So it's not like that we can utilize animals as our uh, own defined methods. Every animal study need to be regulated uh, by different regulatory industry present in the uh, in the world, or in case of India we have CCSEA there, which involves in all these processes. So ethical treatment guidelines is uh, majorly established by CCSEA. Previously, it was known as CPCSEA. So, it established that human uh, human treatment with animals, that good housing condition, good handling practices, best procedure to minimize the pain and distress in animal, those everything required is defined by CCSA. And those defined parameters is always inspected by IAEC, that is Institutional Animal Ethics Committee, which ensure the compliance with all those guidelines that mentioned by CA are uh, worked out by those laboratories which involve in animal research, along with the scientific justification and human procedures towards the animal. And it also complies the validity of research and avoiding the duplication of research, so that the number of animals utilized for the study can be minimized. So majorly, uh, CCSEA involved 4 hours of animal experimentation. That means first is replacement. So if any possible model is available for evaluation of safety of, rather than safety, we can say efficacy of molecules, they replace the experiment with by those in vitro methods if possible. If not possible, then reduce the number of animal, so that we can get a data that is statistically that have a statistical consideration and scientifically or justified for the evaluation. If it is not also possible, then refine the procedure. So avoid the procedures that are inhuman or very uh, cruel towards the animal. And in case of large animals, small animals cannot be rehabilitated, but in case of large animal, rehabilitate those animals, so that they will not pass through sufferings due to other experiments. So after finalizing the after completing the uh, defined experiment, those animals cannot be reutilized for another experiments. So what are exactly CCSA guideline? So firstly, it deals with human care of animals, where everything is recorded and uh, documented towards the CCSA, where appropriate housing condition, appropriate feed and nutrition provided to the animals, appropriate uh, bedding material that utilized, appropriate water supply to the animals, along with that their health management, their clinical evaluation at different time intervals, and which everything will be evaluated by IAC committee during the progress meetings. So basically, it crucially plays important role in managing the ethical principles during conducting the animal research, so that everything is well uh, maintained for considering the ethical statement of the animal. So during clinical trials, we take a consent from the patient that we are giving this kind of treatment to you, and it may have those kind of side effects after administration. So in case of animals, we cannot uh, take a consent from those animals, because of firstly language barriers, secondly they cannot give kind of consent. So in that case, the IIC committee plays a crucial role, where they defines everything is going ethically in considering towards the animal, rather than uh, any abrupt kind of experiments that can be included. And those kind of consent can always be evaluated on the basis of scientific justification of those method, plus ethical consideration towards the animal. So what is basic role of CCSA involved in animal studies is implementation of 4 Rs, registration of animal research facility, so that every experiment conducted in animal research facility can be observed and at every time. So no animal research facility which is not any animal research facility that is not registered with CCSA cannot conduct any kind of clinical trials or selling of animals. So it is basic requirement by CCSA. Then everything related to animal research facility need to be inspected and evaluated, which CCSA does on yearly basis. Along with that, every research protocols that involves animals should be inspected and evaluated before conducting those trials, and which CCSCA controls by using INC committee. Along with that, different nominees are appointed by CCSA to get involved in those scientific discussions, ethical consideration, and uh, evaluation of animal research facility as a research protocol. So CCSA appoint those nominee, and those nominees are well trained by CCSA for evaluation of those parameters, and everything related to import and export of animals is controlled and monitored by CCSA. So what is the basic difference in CCSA and Animal Ethics Committee? So Institutional Animal Ethics Committee is uh, what we can say manufactured or appointed by CCSA, where there are different committee members from which main nominee, link nominee, social aware member, and scientist outside from the institute are appointed by CCSEA, and other team members are appointed from the institute, so that quorum can be completed and every discussion is in ethical consideration along with the scientific justifications for every experiment. So basically, CCSA appoints IAC, and IAC oversees, monitors, and evaluates all animal research related research protocols, institutes, as well as the standard of living of those animal and ethical consideration, and complying those all mentioned parameters by CCSA is compulsory for every animal research facility, and IIC ensure that every institute follows those guidelines. So let's talk about preclinical herbal drug discovery, because at student level we majorly involves in utilizing different kind of herbs or their extract for evaluation of their activities or toxicity or uh, other considerations. So what is the basic uh, baseline that need to be conducted for herbal drug discovery? So first part is identification and collection. So any promising herbal candidate, uh, if we identify them by our literature survey, we can collect those from different areas and dependent time dependently, because their content, phytochemical contents can vary on their different stages of the life. So identification and collection is the most important part for in case of herbal drug discovery. Then extraction and isolation of compounds. So basically, if we can use whole plant as a testing material, then it's okay, but sometimes we need a specific uh, phytoconstituent from that plant, which can be a potential candidate for any specific treatment. So extraction of uh, polarity dependent extraction method majorly utilized, where depending upon the polarity of the molecules, different phytochemicals get extracted and then can be isolated by different chromatographic technique, where uh, we can isolate the fractions, and from fractions we can isolate the single compounds. uh, After that, we can evaluate efficacy of those compound and safety of those compound. We cannot say compound, we can say phytoconstituent, and screen for their different activities along with their pharmacokinetic and toxicological activities. So, first part, on the last, now we have said identification and collection. After that, we need to standardize the plant material. So, first is botanical authentication, that the collected material is the specific species that we have identified. So it will help us to eliminate other species that are close, that are in close resemblance with the target species, that can be eliminated. Secondly, chemical profiling. So we can evaluate different phytoconstituent present in the those plant material, by either HPLC or HPTLC, or but just by evaluating phytochemical constituent. Then along with that, we can evaluate the content by using reference standard. For example, Embolica officinalis is having embellin, so we can evaluate the embellin content present in Embolica officinalis at different stages of fruit, and depending upon that, we can extrapolate the collection timelines for those molecules, and uh, depending upon that, we can have the compound related specific activity in that plant material. So there, like Indian pharmacopoeia, there is a Ayurvedic pharmacopoeia, and it mentions a different standardization parameter for different plant materials, and basically it involves uh, moisture content, ash content, soluble ash content, heavy metal content, microbial load after extraction or before extraction, of after formulation also, and proximate analysis of plant material. So all these kind of herbal based medicines are evaluated using guidelines that are present, which is known as CCRAS guidelines, which focuses on research and development in Ayurveda, and ensures that every study conducted by for Ayurvedic medicine or herbal medicines should be conducted are conducted in scientific and ethical manner. It also defines how clinical trials should be conducted for those Ayurvedic products or herbal products, along with standardization and quality control of those products. So, what are key takeaways from today's uh, presentation? So, first is why in vitro tests are more important. Secondly, what are regulatory requirements that are required for animal studies? Then, what are regulatory requirements for toxicity? How government bodies are getting involved in animal studies, and how they monitor those animal studies? What are preclinical basics for evaluation of plant-based medicines, and what are the guidelines required to evaluate those plant-based medicines? So, thank you all for joining in.
Thank you, sir. Um, it was a nice presentation. You actually took us from initial basic drug discovery, then preclinical, and with herbal drug discovery as well. Um, So there are no questions in the chat box, but just a couple of questions from my side. So you mentioned in vitro anti-aging assay. So what we do there in?
In that case, there are primary cell lines that depending upon our target organs, so either liver or skin or any specific organ. So we induce oxidative stress in those cells by using peroxide, which is a strong oxidizing molecule. And after generation of oxidative stress, we can evaluate the uh, inhibition of those oxidative stress by using our molecules or products, so which will ultimately after at the end all the antioxidant parameters that we conduct in tissues from animal study, all those parameters can be performed and evaluate the inhibition of oxidative stress. So depending upon that, we can evaluate anti-aging potential of that uh, molecule.
So like looking at different radicals which are there in the free radicals, right?
Yes.
One more question. So, like, actually, it's not question. I want to understand your view. So, you mentioned about herbal drug discovery in preclinical. Uh, how do you look, doing animal study on herbal extract or even formulation? Because with the ethnobotanical use of the herbs, the person can directly actually take the powder of the herb, or they can actually even take formulations which are not tested on animal, then again going back and testing on the animal. What actually is the reason to utilize animal for herbal formulation or herbal extract?
Okay, so as we have spoke about CCRA's guideline, the guideline defines exactly what tests need to be conducted for safety and efficacy of the uh, botanical plants. So, if new extract is prepared or any kind of new formulation is prepared, it need to evaluate for safety and efficacy. It is a basic criteria by CCRAS. But if the molecule is already mentioned in Ayurveda, that molecule product mentioned in Ayurveda, that product is utilized for the same Vyadhi as mentioned in the Ayurveda, then there is no need to conduct those preclinical testing. But if you are utilizing the product mentioned in Ayurveda, but for any other different disease, then we need to evaluate those product for the efficacy of that product against that disease. Then along with the safety of uh, that molecule, that product. So
Okay.
CCRA's guidelines gives there is a table they have mentioned where we can conduct depending upon the what formulations we have, what kind of extract we have, and depending upon that we can design our experiment for animal studies.
Thank you. Thanks, Anand sir. It was a nice presentation, and also some of the questions which I feel was important. It was nicely clarified from your side. So thank you for accepting our invite and delivering this preclinical session. Definitely, we will be approaching you regarding the workshop that we are planning next month and also in June. Simultaneously, the work which is going on in collaboration with Solive and Cyro, we will be continuing that. So, thank you, sir. Thank you for joining.
Thank you, sir. Thank you giving me opportunity to speak with students and different members regarding preclinical activities, because everybody thinks that preclinical activities is just animal model, but it is just not animal model. And in case of animal model, there are very differential parameters that need to be evaluated during ongoing experiment. So yes, there is a huge ocean of preclinical models and preclinical testing We have just surfaced it.
Yeah.
Thank you for.
Thanks. Thank you, sir. Thank you.
Thank you.
See you. See you. Good night.
Good night, sir.